Welcome back, friends. In this lecture, we shall be discussing about microbodies or peroxomes. So, microbodies are small spherical organisms bound by a single membrane, commonly found in eukaryotic cell. So, they are small spherical organisms bound by a single membrane. In structure, they are similar to lysosome, and these microbodies they are just like a type of vesicles. So, they appear as ni kama tu lysosome, lakini the difference is their content and what they are doing. So it contains several types of enzymes, but mainly catalase enzyme. So in lysosome we said it contains the hydrolytic enzyme, but here it contains catalase. So we shall go to see what is the function of the catalase. So they are formed by the endoplasmic reticulum and commonly found in the highly metabolizing cells like liver cell and replicate by fission. So they are found in highly metabolizing cell, and we shall go to discuss why in the highly metabolizing cell. Simply, what I can tell you is that during the process of metabolism, there is a chemical produced called hydrogen peroxide. It is written as H2O2. So this hydrogen peroxide, it is dangerous to the body. It is toxic. So the microbodies or peroxomes, they contain catalase. Catalase is the enzyme which can allow the breakdown of this. Under the presence of catalase, the hydrogen peroxide is broken down into water plus oxygen gas. So now, this water and oxygen gas, they are not harmful to the body. But if this hydrogen peroxide could accumulate into the body, it is toxic. So peroxomes, they are very important in highly metabolizing cells because they catalyze the detoxification of the hydrogen peroxide into water and oxygen, which they are harmless. So without peroxomes, we couldn't have catalase, and without catalase, we could not be able to break down the hydrogen peroxide produced in the highly metabolizing cells. So what we are saying is that in animal, peroxomes appear to be confined to the cells of the liver and kidney. In the plant, special microbodies or peroxomes called glyoxomes. These are the special peroxomes in the plant cell. They are called glyoxomes. So occur variety of cells including cotyledons. Peroxomes in the plant cell are often packed within crystallized materials. Now, catalase enzyme containing the microbodies or peroxomes contained in the microbodies. Here is not contained, it is contained. Catalyze the breakdown of toxic byproduct produced from the biochemical reaction within the cell of the organism. The major metabolic toxic byproduct is hydrogen peroxide, as I told you earlier. So when this compound is produced during metabolic activities, since it is toxic, it is quickly broken down into water and oxygen, which is shown as effervescence. So we have hydrogen peroxide here, then it is broken down into water and oxygen. This oxygen appears as effervescence, and this is one among the practical you will do in your Form 6 biology practical. You will have liver cell, and then you have hydrogen peroxide. You will grind the liver, then you will add hydrogen peroxide. What you will observe is the effervescence indicating the oxygen. So why you will see effervescence? Because of the catalase present in the liver cells. Grinding increases the surface area for the reaction of the grinding to take place, of the breakdown of the hydrogen peroxide to take place. So microbodies or peroxomes are numerous in cells of the liver and some seeds of the plant, such as pea plants. So the similar reaction shown by the liver can be seen in the pea seeds, mbegu za njegere. So when a piece of liver dropped into a beaker test tube containing hydrogen peroxide, the liver cells release catalase enzyme, which catalyzes the decomposition of hydrogen peroxide to water and the oxygen gas, which is shown as effervescence. As I told you earlier that this is one among the practical you will do in your Form 6 practical session. So the structure is similar to that of the peroxams. The process of breakdown of the toxic byproduct produced in the metabolism in the cell is called detoxification, because hydrogen peroxide is toxic. So making the toxic material untoxic or unharmful is what we call detoxification. The role of catalase enzyme is to speed up the rate of decomposition of hydrogen peroxide into water and oxygen. So catalase enzyme ina speed up, inaongeza speed kama ambavyo tutasoma kwenye biochemistry, na tutasoma kwenye biochemistry kwamba catalase or enzymes, they are biological catalysts. So peroxide or microbodies in plant are the site of glycolate cycle. That's what we call photophosphorylation, as we shall discuss in the nutrition, how photophosphorylation takes place and how the peroxide, they help in the process of photorespiration. Functions of the microbodies or peroxams. The major function is that they release the catalase enzyme, which catalyze the decomposition of the toxic hydrogen peroxide produced from metabolic process into water and oxygen. The second function, leaf of peroxams are sites of the glycolate cycle. This is not leaf of, it is leaf peroxams. They are site of the glycolate cycle of photorespiration in C3 plants. Also, they are concerned with lipid metabolism by conversion of fats into carbohydrates. Also, peroxams break down very long chain of fatty acids through oxidation, that is beta oxidation, as we shall discuss later in the process of respiration. Note that if catalase enzymes are not produced, or are scant produced, may lead to accumulation of hydrogen peroxide, and therefore an individual may die because hydrogen peroxide is a toxic product. Now, let's look on the plasmodesmata. Plasmodesmata is the singular, plasmodema. Plasmodesmata is the plural, and in singular it is called plasmodema. They are living connection between the neighboring plant cell, which run throughout very fine pores in the walls. This is surface membrane of neighboring cell and continuous underlying the pores. So advantage of plasmodesmata, it enables connection and coordination between cells, since molecules and ions do not have ability to close. They do not have ability to close a cell surface membrane. Movement is required. So they facilitate movement between cell, and plasmodesmata form the sieve plate pores in a sieve tube, as we shall discuss in the transport. So let me just connect this lecture with the lecture of vacuole. Vacuole is the fluid-filled sac which is bounded by a single membrane. It is found in almost all eukaryotic cells. In animal cell, it is relatively small and temporary, such as phagocytic vacuole, pinocytic vacuole, autophagic vacuole, food vacuole, and contractile vacuole. In a plant cell, vacuole is large and permanent, occupies greater proportion of the cytoplasm. In a young developing plant, vacuole is very small, but as plant grows to maturity, the vacuole becomes very large. Small vacuole is known as cuticles. So it is cutiles. So vacuole ni single membrane. Now it is predominant in plant cells, but it's not in animal cell. Animal cell, it have just only small and temporary vacuole, such as phagocytic vacuole and things like that. So the membrane bound the vacuole is known as tonoplast, and the fluid found in the vacuole is called sap. The sap is made up of mineral salt, sugar, amino acid, waste such as tannins and gases such as carbon dioxide and oxygen, and sometimes pigments such as anthocyanins. So vacuole appear just at the vesicles. It has single membrane, which is tonoplast, and has sap. Advantage of vacuole, and these are similar as functions of vacuole. It acts as an organ of osmoregulation and excretion. In aquatic organisms such as Euglena, Paramecium, amoeba, and sponges. So in act kama organ ya osmoregulation, utasikia kuna contractile vacuole. It acts as a temporary storage of waste such as tannins. These may accumulate in vacuole of the leaf cells and are removed when the leaf falls. The tatu occasionally contain hydrolytic enzymes and so perform the function similar to those of the lysosome. That's why I told you earlier that plants say they don't have lysosome, but they have vacuoles which act as a lysosome. In plants, it contains pigment in solution which are called anthocyanins. They include red, blue, purple, and other related compounds. And these, they impart color, which are shaded of yellow and which are responsible for color in flowers. They impart color in flowers, fruits, and leaves. So rather than plastids, we have also vacuole which can impart color in leaves, fruits, and flowers. These may color petals and attract insects for pollination. So without a vacuole, it could be difficult to do the process of pollination. Also, vacuole, they support herbaceous plants. And herbaceous parts of the woody plants by providing an osmotic system which create a pressure potential. They support herbaceous plants. So presence of water in the vacuole create a turbo pressure which support herbaceous plant, and we shall discuss this and see it well in the topics like transport and biochemistry, how water molecules can provide pressure, how water molecules can provide the cohesive pressure, can bond each other and provide the cohesive pressure for the turbo pressure or for the support of the plant. Function as it acts as temporary area for storing material. So can store materials like sugar, glucose, amino acid, organic acid, oxygen, carbon dioxide, mineral salt, and secondary products of metabolism. Vacuole help to increase surface area to volume ratio to help water movement in the cell. So vacuole inasaidia water kuingia na kutoka kwenye cell. Without vacuole, it could be very difficult for water to enter and leave the cell. The eighth function, some of the dissolved substances act as food reserves, which can be utilized by the cytoplasm. For example, we have mineral acids and sugar. Amino acid, they act as the temporary store of the food reserve. So food reserve, it is similar to the sixth one. Waste product and certain secondary products of the plant metabolism may accumulate in a vacuole. So, for example, we have crystals of waste, calcium oxalate, and secondary products like alkalides and tannins may often not action from the consumption by the herbivores. So they may often required or may often be needed by the herbivores. Here the one who wrote it is not seen well because I'm using notes which are written by another guy. But this is the end of the lysosome, and we meet in the next lecture where we shall discuss on the non-membranous structures, starting with the ribosomes. Thank you.